So in the last lecture we looked at some of the methods that neuroscientists have been using to manipulate genes, and in this lecture I want to introduce you to some of the early studies using this method to answer some of the important questions about synaptic plasticity and memory. And then after that we're going to look at some of the more modern methods that have come along recently. So remember that we now have these ways of manipulating genes. We can add or remove genes, so it's called knock-in and knock-out, which affects the whole animal. Or we can use targeted knock-in and knock-out, which affects only some of the cells, and we do that by using these things called promoters, these things that regulate whether or not genes get expressed. And then at the end of the last lecture I introduced inducible knockouts, like tamoxifen in the example that we saw, where you can knock out the gene by injecting the animal with a chemical substance. So you use a promoter so that the knockout will only happen when you add some chemical to that system. And we can use a combination of transgenic mice, so mice that are born with the altered genes, or viral delivery. So if you recall, we looked at how viruses can also be used to get genes into cells. So these are all the methods that we've got. So let's look at some of the techniques that have been used to start to answer the synaptic plasticity memory question. So just to remind you of the plasticity memory hypothesis. So we have been introduced to this idea, the Hebb rule, that synapses should change in strength as part of the memory formation process. And we met LTP, which is the experimental phenomenon that we think corresponds to that. And if you remember the mechanisms of LTP, it involves these specialized receptors on the neurons called NMDA receptors. And NMDA receptors are activated by a convergence of events. One is depolarization of the cell, and one is binding of neurotransmitter to the receptor. So when those two things happen, the NMDA channel opens, calcium flows in. And then the calcium triggers a set of chemical reactions. And one of the enzymes that's involved in that set of steps is called alpha calcium calmodulin kinase. So it's triggered by calcium, and it's a kinase, which is an enzyme that has the job of modifying other proteins, and it regulates the expression of LTP. So if you activate alpha calcium calmodulin kinase, you can produce LTP, and if you what's called constitutively act it, so switch it on so that the cell thinks that it's constantly having to express LTP, then you'll strengthen synapses and keep them that way. Or you can knock them out so that now the cell won't make LTP. So in this early experiment, it was one of the first to use this transgenic technology to look at synaptic plasticity in learning. It's one of the classic experiments. So mice were made that lacked alpha calcium calmodulin kinase, alpha CAMK2. And these mice were very impaired on LTP, so you can see in the graph that the affected mice after the tetanization, the high-frequency stimulation, the evoked potentials didn't really get any bigger at all. And these mice were then tested on the water maze, and they were found to be very impaired in their water maze learning. So they took longer to find the escape platform. Now there's a problem with knockout, the simplest version of the knockout technology, which is that the animals are born with the affected genes. And that means that there's a possible effect on development. And you can see in this slide one of the early attempts to knock out a plasticity gene, the Fin gene. Fin mutant mice, so knockout mice that no longer had this gene. You can see that they had malformed hippocampus with too many cells in the CA3 region. So the normal process of development, which we're going to look at in a few lectures' time, that normally involves some cell death, some sculpting of the cell layers, and this sculpting didn't happen properly, so there were too many neurons. So it's really beneficial if you can use inducible knockout technology, so be able to target your intervention to the time and place of your choosing. So if you recall from the last lecture, there's a way to do this by borrowing from other organisms certain kind of gene systems, and one of them is the Cre-Lox system. So this is taken from a virus, a bacteriophage, and if you remember, the Cre recombinase enzyme made by this virus will hunt along DNA strands until it finds these pairs of LOXP sequences, these two stretches of DNA, and it'll chop out the segment of DNA in the middle. So a mouse that's been transgenically altered so that it has a gene with a LOXP site on either side is called a floxed mouse. And so a floxed mouse, any cell that's got that floxed gene will be susceptible to the actions of Cre. So to be able to use Cre to control whether you knock out this gene, whether you excise this gene, you need to be able to get control of Cre. And we do that using promoters. So in the example that we saw in the last lecture, which is shown in this slide here, just to remind you, tamoxifen was used to control the action of Cre. So the idea is that the Cre has next to it the promoter that's sensitive to tamoxifen. So if tamoxifen is present, that will activate the promoter, that will activate the Cre will cause the gene to be excised. So you're kind of using the promoter as a way of controlling that gene. So this technology was used in an important early experiment to knock out NMDA receptors in a restricted region of hippocampus. So what they did was to make a mutant mouse that has the Cre gene under the control of the CAMK2 promoter. So you remember the CAMK2 enzyme, that's the one that's involved in making LTP. So any cells that produce LTP, in other words hippocampal cells, will, if they are normally making CAMK2, they will also now make Cre. So these mice are then crossed with a floxed mouse, and this mouse has a stretch of DNA which stops a reporter gene being expressed. And the reporter gene, if you remember, reporter genes make some type of protein that can be visualized. So in this case, they're taking LAC-Z, which is a reporter gene. And it's got a little segment of DNA next to it that stops that protein from being made. And that regulatory sequence has LOXP site on either side of it. So normally, that regulatory sequence is active, it's stopping the reported gene being expressed. Now, if that mouse is crossed with the Cre mouse, now you'll get mice that have both the Cre and that floxed gene. Now remember that the function of Cre is to chop out the floxed gene. So the Cre will chop out that regulatory sequence that was stopping the reporter being expressed, and now those cells will express the reporter. So that's a kind of complicated way of saying we're going to show that we can control cells in the hippocampus that have this CAMK2 gene. It sounds very complicated, but if you go back over it, it's all extremely logical. So now these mice are now crossed with another floxed mutant, and this is the one we're really interested in. This is the one with the NMDA receptor and a LOXP site on either side. So if these mice are crossed with those Cre mice that we just saw, that we got produced, now we'll end up with mice that have Cre being expressed. That has enabled the reporter gene to be expressed, and it has also chopped out the NMDA receptors. So the cells that are expressing the reporter will also be cells that don't have NMDA receptors. And you can see in this microscope slide picture here, the little row of purple cells, that's the reporter protein being expressed, and you can see that it's beautifully localized to the CA1 region. So those mice have lost NMDA receptors in their CA1 region. And this was really a tour de force experiment. Nobody had ever managed to control something so precisely like this. And what they found was that these mice don't express LTP, as had been hoped, and was very consistent with all of the pharmacology on the NMDA receptor. So you can see in the middle panel that the mutant mice are not showing that increase in evoked response. And then you can see on the far side the picture of the heat maps of the place cells. So they recorded hippocampal place cells, and you can see that the knockout mice on the bottom there, those heat maps are very broad and dispersed, so they're not nice, fine, tight place fields like we normally see. So these mice with NMDA receptors seem to be a bit messed up in their ability to learn spatial locations. That was the conclusion from this experiment. But really, it was also just an incredible demonstration of how powerful this technology is for precisely targeting interventions to particular regions of the brain. So in the next lectures, we're going to be looking at very recent development, which is using this technology to be able to extremely precisely target in both space and time to exactly where we want.